My name is Jiho, and I chose Jennifer Doudna as my biology role model. Who is Jennifer Doudna? Jennifer Doudna is a biochemist who received the 2020 Nobel Prize in Chemistry for the development of a method for genome editing. She is also a professor at the University of California, Berkeley. She was born and grew up in Hawaii. She majored in biochemistry at Pomona College and earned a PhD from Harvard Medical School in 1989. Growing up in Hawaii, she found herself fascinated by the evolution of natural systems and organisms. She loved math when she was growing up. Even no one in her family was a scientist. Her father gave her a book called The Double Helix, written by James Watson, and that encouraged her to pursue her interest later in biochemistry. What obstacles did she face? She faced obstacles during her freshman year of college in general chemistry course. She even thought about changing her major to French. However, her French professor suggested that she stick with science. Her hobbies are reading and gardening. She likes reading science books, even the books that are not related with her own field. Those are the reasons that I admire Jennifer Doudna. First, she was one of the first women to share a Nobel in the sciences. Second, as a female international student who wants to go to medical school, I was inspired by her passion for not giving up on her dream, even though everyone was telling her that women do not go into science when she was young, in her childhood. Third, she is a professor at my dream graduate school, which is University of California, Berkeley. Those are the two quotes that I like from her. She's involved in the field of molecular biology and genetic engineering. Her research is mainly focused on RNA. CRISPR was discovered as a result of her lab's research into RNA biology. This is the most recent paper from Jennifer. To explain the summary of the paper, conserved phage nuclear cell associated protein that we term CHMC. The researchers suspect that CHMC protects phage mRNA, which encourages the cell infection process by stabilizing the mRNAs and driving their translocation through nuclear cell. If their theory is correct, then cell infection can be stopped by target knockdown of CHMC using mRNA targeting. Here's the figure that shows the result of an experiment on this paper. The figure demonstrates the impact of CHMC knockdown on phage protein levels. After CHMC knockdown, Middle and late expressed proteins were significantly suppressed, while early expressed proteins were not affected much. This suggests CHMC plays a role in non-variant RNA polymerase transcription of middle and late expressed genes. Her research will be still focusing on mRNA and CRISPR. Her lab is currently researching novel mechanisms of CRISPR, CAS, and associated proteins, developing tools for vitro genome editing, developing anti-CRISPR agents, and investigating CRISPR DNA editing.